Okay, so welcome to this next video in which we are discussing the epidermis. Okay, right, so I'll summarise where we've got to so far. So far what we've discussed is the upper layer of the basement membrane of the epidermis. So we've discussed the lamina lucida, as it's called, of the basement membrane, which, remember, is this network of heterotrimeric laminin proteins, okay, which all join together. And we've also looked at how the lower level of cells of the epidermis, known as the basal keratinocyte layer, how these cells are going to attach to the laminin G-like domains of the laminin heterotrimers via integrins which they have on their surface. And we've seen that there are two major examples, well, I've given you two major examples of integrins on the surface of basal keratinocytes. So we've seen alpha-3 beta-1, which is an example of an integrin which will bind to the laminin G-like domain of laminin heterotrimers, and then will link for its cytoplasmic domain indirectly to actin filaments, which are a key component of the cytoskeleton of cells. Okay, right. We've then seen the other example of an integrin, which is the only example of an integrin known, which binds to intermediate filaments. So we've seen the alpha-6 beta-4 integrin, which binds to intermediate filaments, which are another component of the cytoskeleton of cells. Okay, and we're talking about keratinocytes, so the key example of an intermediate filament that you'll find in keratinocytes is keratin. Okay, right. So what we now want to turn our attention to is studying the lower level of the basement membrane. Okay, so let me just draw a picture here. So if this is the basement membrane here, okay, we've got a keratinocyte sitting on top here. Okay, so I'll colour the keratinocyte in turquoise in a moment. So here is the nucleus of the keratinocyte, and it's a basal keratinocyte, so it's one of these in the stratum basale. Okay, and we'll come to the fact that a lot of these cells are stem cells, or cells that have just come from stem cells in a moment. Okay, so this is our basal keratinocyte here. Okay, and then it's sitting on the basement membrane, and as we've discussed, the basement membrane is not just one layer. The basement membrane can be divided up into two layers, okay, like so. One of these layers is known as the lamina lucida, which is the layer closest to the basal keratinocyte, which is the one that we've discussed in depth, and we've seen how this attaches to the basal keratinocytes. And then the layer underneath this is the lamina densa, and we have not discussed the lamina densa yet. So in this video, what I now want to do is discuss the lamina densa, and I also want to discuss how the lamina densa is going to attach to the lamina lucida, which is above it. Okay, right, so what is the structure then of the lamina densa? So basically the lamina densa consists of collagen fibers, okay? So it's made up of a layer of a huge number of collagen fibers. Now, specifically, the collagen molecules that go into creating these collagen fibers are generally type 4 collagen molecules. So we call this layer type 4 collagen. Now that means that the collagen molecules which are going to make up these collagen fibers are going to be type 4 collagen molecules. Now what does that mean? Well, in order to explain that, I need to explain to you what a collagen molecule is, how they assemble into fibrils and then fibers, okay, and then what it actually means to be a type 4 collagen molecule, and then we'll understand what it means to be a type 4 collagen fiber, which just means that most of the collagen molecules that comprise this will be type 4 collagen molecules. Okay, right. So let's start off then with the fundamental protein that we start with. Okay, and the protein is not actually called a collagen molecule. The collagen molecule actually is a trimer of 3 of the foundational proteins which make up collagen. Okay, right. So the foundational monomer of collagen is actually known as a collagen alpha chain. So once again, we're using the symbol alpha. So collagen alpha chain is the name for the monomeric polypeptide, which is actually going to make up collagen molecules. Okay, right, so let's now discuss the structure of these collagen alpha chains. Okay, and before we do that, let me just tell you that there are 45 different genes for collagen alpha chains. Okay, so there are a great number of different alpha chains which you can use to build collagen molecules. Okay, right, so it's a polypeptide then, so here is its amino terminus. Now, the main bulk of the polypeptide is going to be a massive, great left-handed alpha helix. Okay, so let's draw this now. So remember, a left-handed alpha helix is one of these alpha helices which, if you follow it from the top to the bottom, goes round in an anti-clockwise direction. So let's show this. So here we go. I'm coming out of the page towards you. Okay, then I'm going back into the page away from you. I'm coming out of the page towards you, back into the page away from you, out of the page towards you, back into the page away from you. And hopefully you can see that if you follow that round, that goes round in an anti-clockwise direction. Okay, right, so you'll have this very long left-handed alpha helix here. Now I've only drawn a little bit of it, but in reality it goes on and on and on. Okay, so this is the structure of a collagen alpha chain here. Okay, and I'll colour in this collagen alpha chain in pink here. And just to reiterate, it's a left-handed alpha helix. Well, the great length of it is a left-handed alpha helix. Okay, now, there is in fact only 3 amino acids per turn of this alpha helix on average, okay? And there is a general repeating pattern for these amino acids, which I'll discuss in a moment. So this is a left-handed alpha helix. Now, if I draw a portion of this bigger, okay, so let's put a portion here that's much bigger. So here is this left-handed alpha helix. Here is a single turn. Basically, there's only around 3 amino acids in that single turn, okay, shown here now. So we've got 1 here in blue, we've got 1 here in green, and then we'll have the final one here in purple. Okay, and then the pattern repeats again. Okay, so as you go around onto the next turn, which I'll put here, okay, you've got your blue one, your green one, and your purple one. Okay, so we'll put the blue one here, the green one here, and the purple one here. Okay, so it's approximately 3 amino acids per turn, basically. Right, and the general pattern of repeat is that you have a glycine every 3rd amino acid. So let's say that this blue one, this represents a glycine. Now I'll just stress that this is the amino terminus up here, and the carboxylic acid terminus will be down here. Okay, so you have a glycine, then you have an amino acid that's known as the X position, and then the purple amino acid will know as the Y position. Now here I do not want Y to represent tyrosine. Y is the single letter code for tyrosine, but here I'm using Y to represent any amino acid could be there. Okay, but one of the features of collagen is that you have this repeating glycine every 3 amino acids. You have glycine followed by something, followed by something, followed by another glycine, followed by something, followed by something, followed by another glycine. Okay, so if we put in another coil here, you'll have another glycine, another something, and then another something. Okay, so let's just colour this in as well. Here's a glycine in blue here. Here is something in green. Okay, and then there's another something here in vivid purple. Okay, right, and that continues on and on and on and on for this entire alpha helix, basically. Okay, so let me just remind you of the structure of glycine, just in case you have forgotten. Okay, so GLY is short for glycine. Okay, and glycine is the most simple of all the amino acids. So, all the proteinaceous amino acids, anyway. So here is the amino group. Okay, and I'll draw it as a residue, so I'll draw it as though it's within a polypeptide. So here's the amino group bound to the carboxylic acid group of the amino acid prior to it. Then you have the alpha carbon here, with a hydrogen coming off it. Then you have your carboxylic acid group here, which will be linked to the amino group of the next amino acid along. Now the R group of glycine is just a hydrogen atom like so. Okay, so this is the structure of the amino acid glycine, and I should colour it in blue since we've shown it there in blue. Right, so you have this every 3 amino acids basically. Okay, right. Now, which amino acids then do you generally have in the X position and the Y position? Okay, because although I said they could be anything, there are general amino acids that usually you have in the X position, and there's an amino acid that generally you have in the Y position. So, the amino acid that generally you have in the X position, so X is usually the amino acid proline. Okay, so let me just show you the structure of the amino acid proline. So proline is the weird amino acid because its R group actually involves the amino group. So here's the nitrogen, and we're not going to have a hydrogen coming off the nitrogen in the case of proline. Okay, here's the alpha carbon with a hydrogen coming off it. Here's the carboxylic acid group, and now the R group of proline consists of this loop like so. So here's a methylene group, here's another methylene group, here's another methylene group, and then here we've connected that final methylene group up. So you have this 5-membered ring within the structure of proline. So each of these groups here, CH2, this means a carbon with 2 hydrogens coming off, known as a methylene group. Okay, so this is the structure proline. So this is usually what you have in those green positions there after the glycine, which is always there. The glycine is always at that position all the time. Whereas the X position is usually proline, but sometimes you'll have blips and you'll have something else. Okay, now, it's the same for Y. Y, there is an amino acid which generally you have in this position, but it's not a strict rule. It's a generalization, but it's not a strict rule. So Y is usually an amino, whoops, missed the S there, is usually an amino acid known as hydroxyproline, which is not a normal amino acid. It's not what we'd call a canonical amino acid. Okay, but let me show you the structure of hydroxyproline. So basically it's got a very similar structure to proline, as you might have guessed. Okay, so all we're going to do is stick an alcohol group onto proline. So I'll draw this here. So once again we have this nitrogen atom here. We have our alpha carbon with a hydrogen coming off it. We have our carboxylic acid group down here, and then we're going to have our 3 methylene groups. So here's a methylene group, here's a methylene group, and then this carbon here, this is the one that's going to get the alcohol group coming off. So you're going to have an alcohol group coming off this carbon and a hydrogen coming off this carbon. So this is the structure of hydroxyproline. Okay, right. So then, that is the structure of a collagen alpha chain. Now, I've said that there are 45 different genes for collagen alpha chains. Now, they will not all have the same sequences of amino acids, okay? So they will have different combinations of amino acids, basically. They all share the same common structure, that they have a huge, great left-handed alpha helix within them, and that every 3 amino acids they have glycine. But of course, the X and Y's are generally proline and hydroxyproline, but among the different types of collagen alpha chain, the places where you choose not to have a proline in the X position and the places where you choose not to have a hydroxyproline in the Y position will vary. So they will have different sequences of amino acids, okay, although they'll all have this basic sequence where they usually have prolines and X's, but occasionally they'll have places where they don't, but those places will vary depending on which collagen alpha chain you are talking about. And again, for the Y position where we usually have hydroxyproline. Okay, now, these single polypeptides, these are not collagen molecules. They are what are known as collagen alpha chains. Okay, to create a collagen molecule, what you have to do is take 3 collagen alpha chains and combine them together to make what's known as a superhelix. Okay, so let me show this now. So we're now going to show a collagen molecule. okay, which is going to be a trimer of 3 collagen alpha chains, okay, sometimes called a superhelix. Now you might think, well, are we going to create a coiled coil domain? Are we going to wrap the 3 alpha helices around one another? Well, no, is the answer. What we're actually going to do is combine the 3 helices together. Okay, so let me show this. So I'll just underline the collagen molecule in turquoise here. Right, so let's draw one of these collagen alpha chains out, where I'm going to stretch it out a bit, and this will make it easier to understand. So I'm going to, instead of drawing the alpha helix like this, I'm going to imagine stretching it out a bit. It just makes it easier to understand. Okay, right, so we've still got this left-handed alpha helix. Okay, so it's coming out of the page towards you at the moment. Okay, now it's going back into the page away from you, and you see I'm drawing the alpha helix in this different way, but it's the same thing that I'm drawing. I'm just drawing it as though it's stretched out a bit, simply because it's easier to then draw the 3 alpha helices combined together. Okay, so it's then turned around, gone into the back of the page, and it's now coming out of the page towards you. Okay, it's going back into the page away from you, and hopefully you'll see that it's going round in an anti-clockwise direction from the top to the bottom. So here's one of these collagen alpha chains. Okay, now what we're going to do is we're going to combine this with 2 other collagen alpha chains. Okay, so these will be slightly out of sync with it, so I'll put one here, okay, which will just be sort of coming, well, it'll just be out of phase with the other one. Okay, so at the moment it's coming out of the page towards you, it's now turning back into the page away from you, okay, like so, and then it's at the back of the page at the moment. Now it's coming out of the page towards you here, it's then turning back into the page away from you, and it's just slightly out of sync with the other one. And then we'll have another one here, which will then be even more out of sync. So this one will be coming out of the page towards you now. Okay, then it will be going back into the page away from you here. Okay, then it will be coming out of the page towards you now, and then coming back into the page away from you here. Okay, so let's colour them in different colours. So here in red is our first one here. Okay, so it's out of the page at you at the moment. That's folding back into the page away from you. It's coming out of the page towards you. Okay, it's curving back into the page away from you, like so. So that's the one in red. Okay, it's coming out of the page towards you. It's curving back into the page away from you. Coming out of the page towards you. It's curving back into the page away from you. Okay, and then we've got a final one here in green. It's currently out of the page at you. Okay, it's turning back into the page away from you. It's at the moment at the back as these 2 are coming round to the front. Okay, now it's coming round to the front. Okay, and then it's turning back into the back away from you. Basically, we are actually combining the 3 alpha helices together, and that's why this is called a superhelix. It's not a coiled coil domain. In a coiled coil domain, we weren't actually merging the 3 alpha helices together. We were just then wrapping the alpha helices around one another. Okay, this one we're merging them together basically, and this is how you form a collagen molecule basically. Okay, so the question then is, do you have to use 3 identical collagen alpha chains to make one of these collagen molecules, or can you use different alpha chains to make a collagen molecule? Well, the answer is you can use the same, or you can use different collagen alpha chains. So if you use the same collagen alpha chains in the 3 proteins that are comprising this superhelix, that's known as a homotrimer. Okay, homo means the same, trimer means 3-membered subunit. So this is a 3-membered structure where the 3 members are all the same. Okay, if you use different collagen alpha chains in the 3 collagen positions, then it's called a heterotrimer. Hetero means different, so this is a different 3-membered structure. Okay, right, so that is a collagen molecule then. Okay, now, there are 28 different types of collagen molecule. Okay, so these are called collagen type 1 all the way through to collagen type 28. Okay, and the Roman numeral for 28 is 10, 10, and then 8, like so. Okay, so there are 28 different forms of collagen molecule. Now, what determines which type of collagen molecule you are? Well, it's which of these alpha chains you actually use in the 3 positions, basically. Okay, so if you are a type 4 collagen molecule, which is the one that we are interested in, because this is the main type of collagen molecule that we are going to use to make the lamina densa, okay, it means that the alpha chains which you have in these 3 positions here are within the subset of the 45 genes for collagen alpha chains that are considered collagen 4 alpha chains. And there are in fact 6 collagen alpha chains which are considered type 4 collagen alpha chains. Okay, so these are known as the collagen 4A1 gene. Okay, then you have the collagen 4A2 gene, and it goes all the way up to the collagen 4A6 gene. Okay, so basically, I've told you that there are 45 different genes for collagen alpha chains. 6 of these are the collagen 4A1, the collagen 4A2, all the way up to the collagen 4A6. And if you use proteins that are made by these 6 genes here in your 3 sockets, okay, for your 3 collagen alpha chains that are going to make up your superhelix, then your superhelix, your collagen molecule, will be known as a collagen type 4 molecule. Okay, so that's what it means for a collagen molecule to be a type 4 collagen molecule. It means that the alpha chains out of which you make the superhelix are one of these 6 collagen 4 genes, basically. Okay, so they don't necessarily all have to be the same in these 3 positions. You can have maybe 1 col 4A1, 1 col 4A2, etc. Okay, so you can build heterotrimers, it's just that the alpha chains here have to be from this collection of collagen 4 alpha chains, basically. Okay, right, so that's what it means to be a type 4 collagen molecule. Now what I want to explain is what a collagen fibril is and what a collagen fiber is. So we'll start off with what a collagen fibril is. So basically what you can do is you can aggregate these collagen molecule superhelices together, okay, to make a huge great strand, okay, and this huge great strand is known as collagen fibril. So this is not just one collagen molecule. You've taken loads of these things, stuck them all together, and that huge rope that you've created, that's what's known as collagen fibril. Now, collagen fibrils generally have lengths of around a few hundred micrometers. So let's say this one is 100 micrometers, but it could be more than that. It could be 200, 300 micrometers. Okay, and the diameter of a collagen fibril is generally between 10 and 300 nanometers. Okay, so large diameters and large lengths, even larger lengths. Okay, right. Now, that's a collagen fibril. What's a collagen fiber? Well, collagen fibers are the things that you can actually see down a light microscope. You can't see collagen fibrils down a light microscope, but you can see collagen fibers. Okay, so if you've done histology, when you look down a light microscope and it's stained for collagen, you see these sort of swirls. Okay, and those little swirls, sometimes you can actually see individual little strands. Those are collagen fibers. Generally, they'll be stained in a bluey sort of colour. Okay, collagen fibers are then aggregates of collagen fibrils. So you join together many collagen fibrils. Okay, like so. Here's one collagen fibril. Here's another one. You'll join many, many collagen fibrils together, okay, and you'll make something much, much bigger, which is actually visible down the light microscope, and you can continue this on, okay, and that then is what's called a collagen fiber, Okay, so you gradually make bigger and bigger structures. You start with collagen alpha chains, you join 3 collagen alpha chains to make a collagen molecule. Okay, you then aggregate loads of collagen molecules together to make a collagen fibril, and then you can join loads of collagen fibrils together to make a collagen fiber. So the lamina densa is going to consist of absolutely loads of collagen fibers, basically, and they're going to be type 4 collagen fibers, which means that most of the collagen molecules that are actually contained within these things will be type 4 collagen molecules, i.e. the collagen alpha chains will be one of these 6, basically, one of these 6 collagen 4 alpha chains. Okay, right, so that's what lamina densa then consists of, a huge network of collagen fibers, a spider's web, if you like, of collagen fibers, type 4 collagen. Okay, right, what we now want to discuss is how you attach the lamina lucida and the lamina densa together, okay, and this involves studying the proteins known as nitrogen proteins. Okay, so let's draw what we have so far. So these 2 layers of the basement membrane then. Here is the lamina lucida, which I'll just abbreviate to LL, and here is the lamina densa, which I'll just abbreviate to LD. Okay, in the lamina lucida then, we have these laminin heterotrimers. Okay, so I'll draw one of these here, and I'll colour it in. So here are 3 laminin G-like domains in red here, and here is our coiled domain in turquoise, and here are our, these are laminin N-terminal domains rather than laminin G-like domains. I think I just said laminin G-like domains. Then up in orange here, this is our laminin G-like domain here, or the 5 laminin G-like domains here. Okay, right. Now, remember, laminin heterotrimers consist of an alpha, a beta, and a gamma laminin. And you'll remember that I said that generally the gamma laminin of laminin heterotrimers is generally the gamma 1 laminin. Now, the reason this is so important is because the gamma 1 laminin specifically is going to be involved in the attachment of this network of laminin heterotrimers, and I've only drawn one, but we remember that this is a network of laminin heterotrimers. It's going to be involved in attaching the network of laminin heterotrimers, which is the lamina lucida, to the lamina densa, which is this meshwork of type 4 collagen fibers below. Okay, so let's draw one of these type 4 collagen fibers below here. So in blue here, this is our type 4 collagen fiber. Okay, now, basically, there are proteins which combined to the head of the gamma 1 laminin, and they can also bind to type 4 collagen fibers here. So we'll colour this protein in orange here. So these proteins that can do this, and which therefore connect the lamina lucida to the lamina densa, these are known as nitrogen proteins. Okay? And there are overall 2 nitrogen proteins in humans. There is nitrogen 1 protein, and there is also the nitrogen 2 protein, and both of them can do this. Now, for short, the nitrogen 1 and nitrogen 2 proteins are often abbreviated to the NID proteins. So there is nitrogen 1 and there is nitrogen 2. Okay, and both of them can attach gamma 1 and also to type 4 collagen fibers, and therefore they form this link between the lamina lucida and the type 4 collagen fibers within the basement membrane. Okay, so that now concludes our discussion of the basement membrane, which is this most basal layer of the epidermis. Okay, so we now want to work our way up from the basement membrane. So we're now going to turn our attention to the basal cell layer, and the fact that this basal cell layer contains a large number of epidermal stem cells, okay, which are responsible for the tissue renewal of the epidermis. Okay, right, so let's just draw a picture then of the epidermis again. Okay, so let's say this is the epidermis, here are the rete ridges, here's a rete peg, a rete ridge. arete peg, arete ridge, arete peg, arete ridge, arete peg, etc. Okay, now the bottommost layer, as we've spent a lot of time discussing now, is this basement membrane, which now I'm just going to show as a turquoise line here. So this is the basement membrane here. Now, basically, the epidermis is not one constant layer. Okay, you are not just born with your skin and you don't just keep that skin for your entire life. Instead, what's continuously happening is you're replacing the skin. And what happens is that you have a layer of cells right at the base of the epidermis, okay, which I'll draw now. Okay, and I'll just draw it up to here because otherwise it'll take too long to draw it for the entire thing, but it will continue on all the way here. Okay, so you have this basal layer of cells, which are the cells sitting on the basement membrane, and basically in this layer you contain this layer contains the cells which are capable of dividing. Okay, the layers above this layer are not capable of dividing anymore. Okay, now the basic principle of what happens in the epidermis is that the layers right on the top, the layers of cells right on the top, which we'll come to later, those are being shed away into the environment, basically. They are, in fact, what makes dust, basically, one of the key components of dust. Okay? So you're continuously shedding skin cells from the top. Okay? So you also have to be, therefore, replenishing these skin cells. So what happens is this basal layer of cells is dividing to produce new cells, which are then being pushed off the basal layer and are being pushed upwards, basically. These cells go through many different differentiation stages, so they gradually undergo changes, and then once they get to the top, they will then be shedded off. So the epidermis is continuously replenishing itself, it's continuously being replaced. Okay, now, this replenishment is fueled by these epidermal stem cells at the base, basically, okay? Because the cells of the higher levels are not capable of dividing. It's only the cells that are attached to the basement membrane, the cells in this basal layer. Okay, so let's now discuss the cells in this basal layer. So you remember that the basal layer of cells of the epidermis is known as the stratum, which means layer, basal, okay, which makes sense, but it's also called the stratum germinavitum, okay, because of the fact that this is the layer that is dividing and producing the new cells that are going to be moving off from the basement membrane up to the higher levels. Okay, so one of the key components then of the stratum basal is epidermal stem cells. Now, not all of the cells of the stratum basal will be epidermal stem cells, but a good fraction of them will be epidermal stem cells. Okay, so these ones that I'm now colouring in orange here, these represent the epidermal stem cells. Okay, right. Now, what is a stem cell? Well, a stem cell is a cell that is not fully differentiated yet. Okay, and in fact, what's going to happen is these stem cells are going to divide and produce daughter cells, which can then undergo the process of differentiation. And basically, all the way through the epidermis, the different layers of the epidermis, the cells are changing, they're still undergoing the differentiation process, and they only finish the differentiation process once they get to the outermost layer, and then they are shedded away, okay? So, another piece of terminology regarding the epidermal stem cells, okay, they are what are known as unipotent stem cells, okay? Now, what does a unipotent stem cell mean? Well, basically, a unipotent stem cell means that it's only capable of differentiating into one type of cell. Okay, so uni means one. Okay, so unipotent means that it's only capable of undergoing one differentiation pathway. Okay, so although these epidermal stem cells are not differentiated yet, they don't have much choice. They can only differentiate into one type of cell. Contrast that to other terms you might come across. Okay, other terms regarding stem cells are multipotent, okay, pluripotent, and totipotent. So I want to tell you what these three terms mean in terms of stem cells. Okay, so a multipotent stem cell is a stem cell which is capable of differentiating into multiple different types of cell. Okay, so a good example would be the intestinal stem cells, which are in the base of the crypts of Leberkuhn within the intestine. These are capable of becoming different types of cells, basically, because there are different types of cells on the epithelium of the small intestine, and all of them are produced by these intestinal stem cells. So it's capable of producing multiple different types of cell, unlike the epidermal stem cells, which can only become one type of cell. Okay, then pluripotent. Pluripotent is a massive term. Pluripotent means that you are capable of becoming every single cell type within the human body, basically. Any cell, you can become any cell. A pluripotent stem cell is capable of differentiating into a neuron, it's capable of differentiating into a hepatocyte, anything, basically. Okay, any cell of the embryo, basically. Any cells of the embryo. I'll get rid of that. Any cell of the embryo. So what then does totipotent mean? It's like, well, how can you go any more grander than pluripotent? Well, you can. Okay, have a little think and see if you can come up with what totipotent must mean. Totipotent means that you can become any cell of the embryo, but also you can become any cell of the extra embryonic tissue. Okay, so for instance, the cells which become the placenta and the extra embryonic membranes. Okay, so you can become any cell of embryo, okay, and also extra embryonic tissue. Now, very quickly in the developmental process, you lose totipotency. Okay, so by day 5, you've formed the blastocyst, and now the blastocyst is divided into two major types of cells: the cells which are going to become the embryo, okay, which are pluripotent stem cells, and the cells which are going to become the extra embryonic membranes and the placenta. Okay, so very quickly in development, you lose totipotency. You become pluripotent at the very best, not totipotent. But the original fertilized egg cell is a totipotent stem cell, and a few of the cells after that, for instance the cells of the morula, those are also totipotent stem cells. Any cell of the embryo and any cell of the extra embryonic tissues. Okay, so that's what totipotent means. So, as an adult human, we do not have totipotent stem cells. We don't have pluripotent stem cells. At the very best, we have multipotent stem cells. Okay, so any cell of the embryo and any cell of extra embryonic tissue. Okay, so that's just a little bit of stem cell terminology to get the term unipotent in context. Okay, so these epidermal stem cells then are unipotent stem cells. So basically what happens then is these epidermal stem cells can divide. Okay, so if we have one of these epidermal stem cells, I acknowledge that I'm drawing it in rather a different shape from what I've drawn there, but never mind. Okay, so this is one of these epidermal stem cells. Basically what epidermal stem cells do is they will divide. Okay, so this epidermal stem cell will divide into two new cells. So one cell here and another cell here. And these two cells that are produced by the epidermal stem cell are known as the daughter cells. Now, basically, each of the daughter cells faces a choice. Okay, each of the daughter cells can decide whether it wants to remain a stem cell or whether it wants to begin the process of differentiation. Okay, so it's not the case that an epidermal stem cell will just sit on the basement membrane and then suddenly decide that it wants to undergo differentiation process. It doesn't work like that. What happens is the epidermal stem cell will firstly divide, produce two new daughter cells, and these daughter cells then can have the choice. Okay, so they can both make their own decisions. They make their decision as to whether they want to remain a stem cell, okay, or whether they want to go down the differentiation pathway. Now, in principle, both of these daughter cells could decide that they want to remain stem cells, or they could both decide that they want to undergo the terminal differentiation pathway. However, generally, the probability is a half and half that they will decide to remain stem cells or that they will undergo terminal differentiation. So usually half of them decide to remain stem cells and the other half go on to terminal differentiation, and that's good. Okay, that means that we're going to maintain our population of epidermal stem cells, and we're also going to be producing terminally differentiated cells. But it is important to understand that it is not the case that one of them has to remain an epidermal stem cell and one of them has to undergo the terminal differentiation process. They both have the choice, and in principle, both of them could undergo the same fate, basically. So, let's however say that one of them is going to remain a stem cell and one of them is going to go on to the terminal differentiation pathway. So we'll say this one remains a stem cell. So it's going to make the choice that it's going to remain a stem cell. Okay? It will therefore remain attached to the basement membrane, and if it wants to now undergo differentiation, what it will firstly have to do is it too will have to divide into two cells, and these two cells then have the choice of whether they want to undergo terminal differentiation or not. Okay, let's say that this one here, on the other hand, picks terminal differentiation. Now, when it has picked this terminal differentiation pathway, it does not instantly say, right, now I'm off, and it doesn't instantly detach from the basement membrane and move upwards. Okay, we know it can't do that because we've got holes here. What are these other cells? Okay, these other cells are the ones that are on their way to becoming terminally differentiated cells. Okay, it takes time for a cell to completely detach from the basement membrane and move upwards. It's not an instant click of the finger thing. Okay, so they remain attached to the basement membrane for a good period of time. Okay, so it's going to undergo the pathway of terminal differentiation. Right, so what I want to stress is that terminal differentiation means that you are going to change your characteristics hugely. This cell is going to undergo a lot of changes. You can compare it, if you like, to undergoing puberty. It's going to undergo a lot of changes. Okay, now this does not happen with a click of the finger. Okay, so it takes time. There is a lot of time between it going from being a daughter cell of an epidermal stem cell to being a terminally differentiated keratinocyte. Okay, right. Now, one of the early stages of this differentiation process is that it's going to become something known as a transit amplifying cell. Okay, now these transit amplifying cells, these sit on the basement membrane of the epidermis, okay, and they do exactly what their name says. They amplify. Okay, transit just means that they are in the transition from a daughter cell to a terminally differentiated keratinocyte. Okay, amplify means that they are going to amplify their numbers. They are going to divide and divide and divide. Okay, so we will colour in this transit amplifying cell in blue, and this is what these other cells of the basal layer are. Okay, these are transit amplifying cells. Okay, so what they are is they are daughter cells of epidermal stem cells which have decided that they are going to be adventurous and they are going to undergo the terminal differentiation pathway. Okay, now, they are only in the very early stages of differentiating, okay? And at this stage, they are still attached to the basement membrane, and they are still capable of dividing at the moment. And this is what they're going to do. They're going to divide and divide and divide, produce more transit amplifying cells, and all of the cells that they produce. Okay, so let me show this. So we've got one transit amplifying cell here. Okay, and it's going to divide into two transit amplifying cells. All of the cells that this transit amplifying cell is going to produce will be in the same state as the original cell, i.e. they will all be now committed to undergoing the differentiation process. So it's not like, one of the daughter cells of this transit amplifying cell can suddenly decide that it would like to become an epidermal stem cell. It can't go back now. Its mother had taken the decision that it was going to differentiate, and now the two daughters are committed to this. Okay, so both of these daughter cells are also transit amplifying cells on their way to the terminal differentiation process. Okay, so this is just basically an amplification of the number of transit amplifying cells that you have. Okay, so it's just a way of increasing the number of terminally differentiated cells that you can produce from this population of transit amplifying cells without the need for the stem cells to do more work and generate more transit amplifying cells. Okay, right, so now what's going to happen is as you continue through the differentiation process, one of the things which will happen is the transit amplifying cells will now detach from the basement membrane. Now, when they detach from the basement membrane, they have undergone enough changes now that they are no longer capable of dividing. Okay, so in these higher levels now, there is no division. Okay, so let me reiterate that. In the basal layer, we've got these transit amplifying cells which are capable of dividing. Now, as they progress through the differentiation process, they will get to the point where they can no longer divide. Okay, so let's say it's here. And at around that same point, they will also detach from the basement membrane. So this is the point where they're going to detach, and around that same time they lose the ability to divide, which means that the cells of the upper layers of the epidermis are not capable of dividing. Okay, so let's now talk about the next layer of the epidermis, or in fact the next layers, because it's multiple layered, the next layer that's given a name. Okay, so the next layer that is given a name, okay, is what's known as the stratum spinosum. Okay, that's sort of like its older, pretentious name. Okay, many people would just call this the spiny layer, or also the prickle cell layer. Okay, so stratum spinosum, layer spiny, the spiny layer. Okay, so many people would just call this now the spiny layer. Or people might even call it the spiny layers, because there are actually two layers of these spiny cells. Okay, right, so let me show this. Or, well, actually I don't know if it's necessarily two, it could be more than that, okay, but there are definitely more than one layer of these spiny cells. Okay, right, so let's draw these here. So you have these layers of cells which have just come off the basement membrane. Okay, now why is it called the spiny layer? Well, it's because these cells look prickly. Okay, so all of these cells are what are known as spiny cells, or they're also called prickle cells. Okay, and I want to now explain why they appear prickly, why they appear to have spines. Okay, so we want to take out one of these cells of the spiny layer, okay, which I'll highlight in pink. I'll highlight all of these prickle cells in pink here. Okay, we want to now take out one of these cells and see why they're called the prickle cells, why this layer is known as the prickle cell layer, and why it's also called the spiny layer. Okay, so basically, if you look at one of these cells, and this is underneath the light microscope, you can see these things. You see these large sort of spines or prickles coming off them, these large sort of dark portions, like so, which are connecting to other cells. Okay, so you'll have another prickle cell here. Okay, and basically these are known as the prickles of these prickle cells, and this is the origin of their name. Okay, so this layer, stratum spinosum, has very obvious structures that resemble the prickles of some sort of cactus or some thorny plant, basically. Okay, right. Now, what are these structures? Well, and what I've suggested here is that it's some sort of contact between neighbouring cells, and that is indeed correct. Okay, these structures are known as desmosomes. Okay, right. Now I think we'll call it there for this video, and in the next video what we'll do is explore the structure of a desmosome, okay, which is this cell-cell adhesion structure. Okay, so it's a way that you can adhere two cells together, and it's made use of in this spiny cell layer, and they are very, very evident in this spiny cell layer.